This presentation will discuss the validation of an updated next-generation sequencing assay for myeloid neoplasms. To get started, I'd like to talk about what is next-generation sequencing, or NGS. NGS is a molecular assay that allows us to determine the specific nucleotide sequence of DNA. This DNA can be acquired from whole blood, bone marrow, as well as formalin-fixed and paraffin-embedded tissue. Once the DNA has been extracted from the tissue, it will be fragmented into smaller, easier-to-handle portions. An adapter will be added, allowing the DNA to adhere to a flow cell, remain in one location, and be amplified. Fluorescently labeled nucleotides will be added to the growing DNA strand. As these nucleotides are added, they will give off a flash of light whose color is correlated to a specific nucleotide. This will allow us to determine what nucleotide was added. After all the fragments have been sequenced, they will be pieced back together to recreate the gene using a reference sequence as framework. One benefit to NGS is the high throughput sequencing, meaning millions of fragments are sequenced simultaneously. This allows us to sequence numerous genes at one time, and we can use this to create panels of multiple genes that are associated with a disease or group of diseases. Previously, we have utilized the TrueSight assay to sequence genes with known involvement in myeloid neoplasms. If you look to the left, you will see the gene that is being sequenced, and on the right is the portion of the gene sequenced. While some genes will have the entire gene sequenced, others will only have certain exons that are sequenced. This is due to these genes having what is called a hotspot, which is an area that mutations happen time and time again. It is therefore more efficient to only sequence this area. While the assay has helped us greatly in the past, there were some additions that we wished to make, which is why we have chosen to switch to the SOPHIA assay. The SOPHIA myeloid panel has an overall similar gene content to TrueSight, however there are a few exceptions that we have highlighted here in red. The first gene I'd like to talk about is the CEBPA gene. This gene has a high GC content, which means there's a higher number of hydrogen bonds when compared to areas with more AT. This makes sequencing by NGS difficult and therefore has required ancillary testing by Sanger sequencing. The second gene, FLT3, contains internal tandem duplications, meaning a portion of the sequence is repeated multiple times in a row. This adds complexity to the sequencing and can provide challenges with bioinformatic mapping. Therefore, supplemental fragment analysis has been required. One of our goals with the new assay is to improve the coverage of these genes and therefore eliminate the need for the supplemental testing. We also wanted to add two new genes to our panel. The first is DDX41. This gene has numerous roles, including innate immune sensing, RNA metabolism and splicing, and tumor suppression in hematopoiesis. This gene has been implicated in myelodysplastic syndrome as well as acute myeloid leukemia. It is a germline mutation and it is inherited in an autosomal dominant pattern. We wanted to add this gene to our panel because it has been found to be the most common germline mutation implicated in myeloid neoplasms. The second gene, UBA1, which is involved in the initiation of the ubiquination process, meaning it marks proteins for destruction. This gene has also been implicated in AML and MDS, as well as lymphoid neoplasms. The mutation in this gene arises in a somatic fashion. Due to this gene's involvement in VEXAS syndrome, it was actually Carmanos who reached out to us first to imply about adding this gene to the panel. So now that we had our panel, we needed to ensure our test was correct. This led us to assay validation. Assay validation is essentially asking, is this the right test? It will help establish performance characteristics, including accuracy, precision, sensitivity, specificity, and the limit of detection. It will also ensure that the test meets clinical requirements. This is required in specific situations, including laboratory design tests, such as ours, modified FDA-approved tests, and new methodologies. In addition to the SOPHIA panel, we also are using the SOPHIA DDM bioinformatics platform for secondary analysis. This software will align the test sequence with the reference sequence and identify the variants between the two. It will then filter out the variants and report those that are classified as pathogenic, likely pathogenic, or a variant of uncertain significance. As a part of the validation process, we needed to determine the accuracy of the test, meaning, is the test detecting the variants that are present? 201 variants were detected by the assay, including 127 single nucleotide variants, 47 insertions or duplications, and 27 deletions. When comparing this detection, as well as the variant allele frequency, to prior assays and reference information, there was a 100% concordance for all genes with a variant allele frequency of greater than 5%. To better understand what we mean when we say variant allele frequency, we are stating how many alleles are present in the sample that contain the variant as a percentage of all alleles present. For example, if there are 100 alleles in a sample and 30 have the variant, then the variant allele frequency would be 30%. We also needed to determine the precision and reproducibility of the assay. For this, we used 3 samples that have previously been tested as well as a control sample, HD829. These samples were run 5 times in total to determine the intra-run reproducibility, which occurs between the same run, as well as the inter-run reproducibility, which occurs between different runs. Our results showed there was a 100% concordance with the expected results, meaning that the Sophia test reproduced similar results each time. In the charts below, you can see some example data from the TrueSight assay on the left and the Sophia assay on the right. These charts show that not only was the Sophia assay able to detect multiple variant types, it also did so at a similar variant allele frequency. For example, the IDH1 gene here had a mutation that was picked up at a variant allele frequency of 27%. The Sophia assay picked up the exact same mutation at a variant allele frequency of 28%. This means that the Sophia assay is reproducing concordant results with the TrueSight assay, allowing us to determine 100% concordance with expected results. We also wanted to determine the analytical sensitivity and lower limit of detection. We had a clinical reporting cutoff of 5%, meaning the specimen must have a variant allele frequency of at least 5% to be reported. To evaluate this assay and make sure it is sensitive enough to maintain that 5% cutoff, we used the control sample HD829 in 3 replicates across 3 runs. This sample is known to have 22 variants that range in variant allele frequency from 5 to 70%. Our assay was able to detect 100% of these variants, showing that it is sensitive enough to maintain that 5% cutoff. We also wanted to determine the lower limit of detection, which is the point at which the test can no longer consistently detect a variant. This is beneficial when the variant allele frequency needs to be reported in post-treatment samples. To determine the lower limit of detection, we performed a 50% dilution and once again ran 3 replicates across 3 runs. We were able to detect 21 of the 22 variants at a 2.5% variant allele frequency. FLT3 was also detected at a 2.5% variant allele frequency. However, this was only in 2 of the 3 runs. Remember though, FLT3 is very difficult to sequence in the first place by NGS. In summary, our assay had a 100% concordance with greater than 5% and an almost 100% concordance with a greater than 2.5% variant allele frequency. Overall, what does this assay do? Well, first, this assay improves our gene coverage. All genes that we want covered are covered with a greater than 5,000 times depth. A higher coverage helps points to a lower risk of a false negative. The FLT3 gene, due to hybrid capture assay and improved mapping, was able to be detected down to 2.5%. We also updated our gene content, adding the DDX41 and UBA genes. We were also able to complete gene sequence with additional genes as opposed to their hotspots, which allows for improved diagnostic yield. We were also able to improve our laboratory workflow. We eliminated the need for the CEBPA supplemental sequencing due to the hybrid capture assay improving its coverage. CalR supplementary fragment analysis was no longer needed due to the improved next-generation sequencing coverage of CalR. We also were able to eliminate the need to repeat samples to resolve low-frequency variants due to the improved lower limit of detection as well as the reduced noise. We also improved the turnaround time due to the elimination of the Sanger sequencing and supplemental fragment analysis. This also allowed technologists time to be freed up to the point of 0.7 full-time equivalents were freed for technologists, allowing them to work on other projects instead of one long project. We also were able to improve our costs and savings because less reagents were needed with the elimination of those additional tests. Overall, we were able to properly validate our test and show that it is accurate, precise, and reproducible. We were able to improve our gene coverage as well as introduce new genes. We were able to cut down on additional tests as well as improve workflow. Overall, this assay will help improve our molecular diagnosis of myeloid neoplasms. Thank you for watching.